Hi, and welcome to the fifth webinar in our Horizon series. This series is meant to provide an easy introduction and credible next points of reference for the big things facing Ontario livestock sector. These big things will determine whether or not the Ontario livestock industry has a robust future. Today's session breaks from our normal noon hour time slot because today we welcome a speaker from Australia. Each Horizon series webinar is complemented with a white paper co-authored by LRIC and a well-known and respected expert in the topic area. You can find all of the white papers and recordings of the webinars on our website, livestockresearch.ca. During the presentation, please submit your questions using the Q&A function, and at the end, Jean Howden will bring them forward for discussion. And now, I'll ask Chloe Neudorf, our research assistant, to introduce today's speaker. Enjoy the session.
Thank you, Mike. Today, we are pleased to welcome Dr. Stephen Miller, Deputy Director of the Animal Genetics and Breeding Unit in New South Wales, Australia, with the University of New England. Prior to his work as Deputy Director, Dr. Miller worked at Angus Genetics Incorporated as a Director of Genetic Research. Dr. Miller is a contributing editor to the fifth white paper on genomics and is here to speak on that topic. A University of Guelph alum, Dr. Miller has published over 100 peer-reviewed papers and is recognized for world-class research. We are pleased to have Dr. Stephen Miller as LRIC's genomics expert.
Thanks, Chloe, for that introduction, and thanks, Mike, McMorris, and Jean, and the team at LRIC for this opportunity to talk about genomics today. It's a privilege to present on this topic, but also it's kind of nice to talk with I guess producers and other people that are interested in the future of livestock in Ontario. I grew up on a cow-calf farm not too far from Guelph, just outside of Cambridge, and spent my undergrad and graduate years at Guelph and about 14 years on faculty there. So it's in a way, it's kind of like coming home to give a webinar. But today I'm here in my office at the Animal Genetics and Breeding Unit here in Australia, and I guess that says something about COVID in Australia, because we are pretty much back at the office. This is, I thought I'd give you a little bit of idea where exactly I sit. And we're just between Sydney and Brisbane on the East Coast, and about a couple hours in from the coast, but we're up a little over 1,000 meters. So it does get cold here, although we're at the same coordinates. If you look at the, I kind of looked up our longitude from the equator, and it's very similar to Jacksonville, Florida. And if you're in Ontario, you're thinking, well, Jacksonville, Florida, that's pretty nice. But the forecast today was for snow, and it's again forecast tomorrow for snow, but that is totally uncommon around here. In fact, people will probably go out and take pictures and things because it doesn't snow here that often. But we do get 4 proper seasons here in Armidale, which is not typical for Australia. Just reflecting back on and it has to do with genomics and kind of where the technologies got to in terms of livestock. But when I was starting out as a graduate student in animal and poultry science at the time in Guelph, I was approached by another colleague, so he's been around for a while, in the basement of animal and poultry science. And he didn't exactly say, go west, young man, but he basically said, go east, young man, go across the street, go across Gordon Street to molecular genetics department. That's where the future is. Don't be, we're just getting to the point where people are discovering genes, and that's going to be the future. And you're not going to be really have a future with the way we do breeding here in animal science anymore. Well, I didn't take that advice to heart. I stuck there in animal science in the Center for Genetic Improvement of Livestock, and glad I did, because he was only partly right. That molecular genetics, it turns out, is the future, but the application of it isn't at all what he had envisioned at the time. What I've got here on the left is a picture of what we call a Snip chip, and they've been around now since About 2005 is when the first generation of this particular technology was really implemented for livestock, but it didn't happen overnight. And I'm jumping a decade or so of activity between when I was told to not pursue it, livestock genetics in animal science, but to go over to molecular genetics into 2005. Things moved along at a pace where we started to discover some genes, and we started to discover mutations in those genes that affected some traits in livestock, but it wasn't hugely successful the way the technology that I have on the screen is today. This was really transformative, and this followed a sequencing of the human genome. Then they sequenced the cattle genome, and once they sequenced the cattle genome, they identified a whole bunch of variants in the genome. But there is also this technology, and it's simply it looks like a glass slide, and you'll see 96 little squares on there. But each of those little squares is where we can genotype an animal. For today, in 2005, it was 50,000 SNPs, or single nucleotide polymorphisms, or differences in the DNA for an animal across the whole genome. And so we started to call it whole genome selection. So that chip came out in 2005, and it was really the first time that we were able to genotype large numbers of animals for that large number, what we call moderate density or even high density, 50,000 SNPs. Before that, we were doing, a handful of SNPs, 5, 10, and suddenly to jump to 50,000 across the whole genome allowed us to implement today what we call genomic selection. And that has really transformed livestock breeding since 2005. When it first came out in 2005, the question was, can we make it work in livestock? And in 2009, the dairy industry basically implemented it to great success, and the beef industry was looking, saying, hey, can we do the same? And in 2021, I'm here today to say that all the above today that is all history, and this technology is being used in a lot of species across livestock. I have a picture here of, I was very fortunate at Guelph to be work with some great people, to have some great faculty teaching me. And this is one of them, Dr. Larry Shaffer, who's still active in the field. I wouldn't say retired. I think officially doesn't pull in the paycheck anymore, but I understand he's still very much active in animal breeding. But he, shortly after that chip came out in 2005, before it was being really implemented, he wrote this paper in 2006. And what he predicted there was we could use this chip to basically double the rate of genetic change in dairy cattle and greatly reduce the cost of testing bulls. And what we've seen in practice is that Larry was exactly right. And that's of no surprise if anyone listening knows Larry. So what this graph is from Lactanet, Canada, and it shows the increase in the Dairy Genetic Evaluation Index, Lifetime Profit Index, which kind of combines all the traits into an index for selection. And up until Genomic Selection was launched in around 2009-10, the annual rate of gain for dairy cattle was about 46 points a year on the index. After that, with the implemented genomic selection, they're now doing over 100 points. So a little over doubling the rate of genetic gain, actually. And there's the implementation of genomics, but there's some other that allowed them to tackle some other traits like health and fertility and that in a better way as well, which increased the gain. But overall, Larry's prediction was correct. And really what the technology is doing is it's allowing you to come up with an accurate genetic prediction based on the genotype. So we can actually genotype young bulls, and we don't have to wait until that bull has 70, 80, 90 daughters in milk when he's a number of years old at that stage. So it really shortens the generation interval and increases the accuracy on selecting those young animals. I've got a picture of a car maybe I'd like to own someday, a Porsche 911. Because I think back in 2000, let's say 2005, before the chip came out, you'd hold up the International Holstein as kind of one of the poster childs of genetic improvement. It was the race car of progress. And we've just doubled the speed or doubled the acceleration. Imagine this Porsche here that can do 0 to 100 Ks in less than 4 seconds. Well, imagine now you can do it in less than 2. That is essentially how much faster now the dairy industry's making progress for profitable traits in the industry for farmers based on genomics. It's quite incredible when you think about to double anything really. So pulling back to the beef story, in 2010 the question was, can we implement the same thing in beef and have success like we did in dairy cattle? Well, the answer is yes, and it took a little longer. It was a bit of a chicken and egg in beef cattle, where we saw if we had an accurate genomic prediction and people started to use it, it would actually eventually it kind of fulfills itself, because all the animals are getting genotyped. Those animals then go on to have phenotypes, and it drives that reference set that we need to come up with accurate predictions. And so this is just a graph of genotyping in the North American Angus evaluation. So I used to work with American Angus, still do some work with American Angus. And the genetic evaluation there, when I first started in 2016, they had just over 250,000 genotypes. And that was phenomenal, really, to consider in a single breed to have that many genotypes. This year, they will hit 1 million genotypes. They're doing just over 165,000 genotypes a year, which is over about half of all calves registered. A little over half now are genotyped. It's just becoming a routine part of the business for farmers, because the accuracy is there. They're getting, and we've done studies to show what the predictive accuracy is based on genomics, and it's more accurate now to have the EBVs or the EPDs or genetic evaluations. The predictions that we create on the animals is more accurate now with simply just a genomic sample than if we went out and measured the actual animals for the trait that we're interested in. So take weaning weight, for example, where we can predict that animal's genetic merit for weaning weight more accurately with just a DNA sample at birth than if we waited and actually weighed that calf at weaning and used its parents and its pedigree and everything that we would normally do. So it is the technology is transforming the way breeders breed their cattle and do business. So those are cattle examples, but the technology is not restricted to cattle. It's available pretty much to all livestock, all animals really. We're doing projects here at the Animal Genetics and Breeding Unit in honeybees, in they call prawns or shrimp, the ones you might want to eat someday, and plants and other species as well. But in terms of livestock, this is a livestock webinar. Pretty much any species that you want to deal with, we could apply genomics. And this is Sheep Genetics is a product that the software and tools are developed here at the Animal Genetics and Breeding Unit, and genomics is a big part of the rollout of breeding values now in Australia for a number of traits. And this is just to give you an idea of the spectrum of traits. Basically, what all the things that are measured in the early days, you'd think about, well, we're interested in growth and we're interested in wool. We might add carcass traits, and there we have there's actually over 70 different traits that are evaluated in the sheep genetic evaluation here in Australia. Carcass traits includes a lot of eating quality traits, reproduction, number of lambs born, number of lambs weaned, lamb survival, those types of traits. And then we get into, on the bottom here, some bit more special traits. And I think if we think about what's the role of genomics, and Mike said about, what's the sustainable future for livestock, there are some traits that are a lot harder that we haven't been able to tackle very well. Like we have growth, for example. Well, we could measure growth on the animals, on our selection candidates, and we made good progress even before genomics. But some things like health are a lot harder because The picture here for health, that looks like a funny picture. And if you didn't know sheep breeding, you might wonder, what is that exactly? That's the back end of a sheep, but it's actually the back end of a very desirable wool sheep because it has an open breech and has no wrinkle. And the idea there is you want a nice clean back end on your sheep, because dirty back ends on sheep here in the New England, where there's a lot of fine wool production in Australia, they get what they call fly strike. And basically that is bad for the sheep. And what we can actually breed for a healthier sheep by addressing that through breeding value. So we actually breed for less breech, what they call breech wrinkle, in the back end of the sheep. So it's less wrinkly, it's more open, and a cleaner sheep is a healthier sheep. This here, sheep genetics and WEC, which is worm egg count. So as all farmers would know, if you've got cattle or sheep or whatever out on pasture, internal parasites are a problem, and there's particularly nasty worms in this region that will kill sheep. They will become anemic and they will die. But you can count the eggs of those worms in the feces of the sheep, and you can actually select for sheep that have a better resistance to worms. So through genetics and genomics, we are selecting sheep that are healthier, require less chemicals and less potentially surgical interventions to stay healthy. So, in genomics basically just accelerates that and makes that better and makes us go faster for those traits, because some of these traits are hard to measure, but we can measure them on what we call a reference group of animals, and then that prediction then works for other animals where we don't have the luxury of being able to take the measurements. But it points to a very important aspect. I think that's important for everyone to realize, that genomics is not magic, that it's still a lot of hard work. It requires animals that have the measurements that you want to change. So we need that reference set, literally thousands of animals that are measured for the trait and genotyped and related to the candidates that we need to select. So it's not a magic bullet that if we just start genotyping our sheep and we can select for anything we want. You actually need that reference set of animals measured for the traits, as well as genotyped. Another example closer to home there in Ontario, hybrid turkeys. This is a clip from their website, and a picture through the courtesy of Dr. Ben Wood. But genomic selection is being applied now in turkeys, hybrid turkeys there in Kitchener, part of Hendricks Genetics, working with colleagues at the University of Guelph to roll genomic selection out for the turkey breeding program. It also indicates here, they've been using it in their pig and layer programs ahead of turkeys. But again, every species, if advanced breeding programs where you have lots of animals measured, you can develop the chips, do the genotyping, and basically increase your accuracy. The USDA published a blueprint for animal genome research a couple of years back now, and they basically highlighted a lot of what I've talked about so far. The success in a lot of species, that the application of genomics and how it's been transformative. But then it looks to the future as to what type of research and what are the areas we need to focus on. Those hard to measure traits, the health traits that we just talked about in the sheep, for example, matching animals to environments in terms of extensive livestock. They're working over a range of production environments, and some genetics are going to match those better than others. We're going to talk about sequence data here in a minute, but a lot of in that white paper, in that blueprint, is how do we actually use all the sequence data that we will have available and do have available on animals, so we can drill down to what we call the causative variants. What are the mutations in those animals that's causing increased milk production or better resilience to worm burdens or whatever that trait may be. In some ways, we're coming full circle back to my advice to go across to molecular genetics in the early days. Getting back to now, okay, maybe we're getting to the point now where we can actually start to better identify and use these causal variants. From that blueprint, USDA blueprint, just another example to show that we're not just limited to our terrestrial species. This is rainbow trout, but showing increased accuracy with genomic predictions, and they basically doubled their accuracy. And to go back to Larry Shaffer's prediction, you're going to double your rate of genetic gain. Rate of genetic gain is directly proportional to accuracy of selection. So if you double your accuracy, you double your gain, and it seems to be holding up in trout as well. So we have the technology that's being applied, say in Angus cattle, dairy cattle, sheep, fish, turkeys, pretty much you name it now for the more developed species. What are we going to focus on now? And everywhere I go, or as you talk to different groups, the focus is really on, we've made a lot of ground up to now in terms of what are the easier traits to measure, say growth rate in cattle, or carcass traits, or milk yield, et cetera, in dairy cattle. But there are some traits we would have always liked to focus on, but they were always really too hard without genomics, because we could measure these things on some animals, but those weren't necessarily our selection candidates. Things to do with health, adaptation, fertility, longevity, robustness, and resilience are kind of that same set of traits, but we want animals that really thrive in the environment without the use of chemicals or other management interventions that might allow that. Just to give you one example of that is a project we had with American Angus, and that was looking at adaptation to heat stress. So this is just a map of the US and a heat map, and I often use that in places like here in Australia to show them that it does get hot in places other than Australia. And where I was in the US was right here in Missouri, and I can say it was more humid than Southern Ontario for more of the year. So it is, heat stress is an issue. It's especially an issue in the US because all through this region here is what they call the fescue belt, and they have a toxin in the grass. It's basically the grass is everywhere. Most pastures are fescue because it grows really well, but it has this toxin which affects the cattle in a negative way, and that they can't dissipate heat like they normally would. It affects their it affects their ability to vasodilate and dissipate heat. So what we did is we identified what we call a hair shedding EPD, and that allows to identify cattle that shed their coat better, and that's highly heritable. And we rolled out basically a genomic prediction for hair shedding, and that allows cattle to, and this was in conjunction with University of Missouri, but this is now based on about 14,000 scores, allows breeders to actually select those cattle that are slicker coated and more adapted to heat. Just gonna have to go a little bit quick here, but this was a press release that came through detecting SARS in wastewater using genomics, and I've got circled here Lumina. Lumina is the company that developed that first Snip chip. And that Snip chip didn't come about, Lumina didn't say, hey, we can do a lot in livestock. It was really developed for humans, and it's been applied for lots of applications. So all this work that Lumina is doing and other companies now are doing in terms of sequencing, even just this morning, I heard a blurb on the radio about identifying the variants that are showing up and genome sequencing. Everybody knows about genome sequencing now. And all those technologies that are being employed to basically speed up COVID detection will speed up also how we apply sequencing in livestock. And I haven't actually, I'm going to use this as an example to show the difference between sequencing and SNP chips, but I've put up here the the words to O Canada, and if all of those words was the sequence on an animal, there's about 240 characters there in O Canada, and a SNP chip has about 100,000 SNPs on it today, but with sequence we have in Angus anyways, we have about 20 million SNPs. If we were to sequence the animal, we would identify 20 million SNPs. So with a SNP chip, we're only picking up about 1 in 200 SNPs. So if I was to, if this O Canada is sequence, the same version in a SNP chip would look like this. We'd only pick up about 2 letters of O Canada if we were to use that same ratio. Basically, the T in north and the G in the last guard for, stand on guard for thee. So you might look at that and you say, well, Steve, you say these SNP chips are fantastic, but that doesn't look like a very good rendition of O Canada. Looks a little sparse, but it's amazing how well, I've showed you how well it actually does work, because it works very well. And why does it work so well? Well, I'll use an example here, compare it to the Australian National Anthem. So I'm going to compare the Canadian Australian National Anthem, and the Australian National Anthem is about twice as long, so I've just taken the first half of it. But I lined up the Canadian National Anthem and the Australian National Anthem. And when I lined them up word for word here, I think around letter 98, we have the T for North in O Canada, and there's a D in the Australian National Anthem. And at the other position, it's a G in the Canadian National Anthem and the I. So if somebody was just to feed me and I had this genotyping tool that only genotyped at those 2 positions in the National Anthem, and they fed me the script for any national anthem, American, Canadian, whatever, and I got a T and a G based on my reference, I'd say, oh, that's the Canadian National Anthem. If I got a D and an I, I'd say, based on my reference, I'd say, well, that's the Australian National Anthem. And based on that information, I could actually just fill in the rest of the words, because I know if I got the D and the I, when people pass out song sheets, they don't really mix them up. They don't pass out half of the Canadian, half of the Australian. So, and when we inherit DNA, we basically inherit in big chunks, just like, the entire words to O Canada, or the entire words to Advance Australia Fair, which is the Australian National Anthem. We're using a technology here called imputation, and imputation allows us to infer dense information based on sparse information. And this is being used now with, and this is why I talk about sequencing, because there are companies like this company here, Genco, and doing some work in cattle and showing that they can basically deliver for the same cost of a SNP chip now, not 100,000 variants, but actually the entire sequence for about the cost of a SNP chip. And I see this is the next step change that happened in 2005, where we got 50,000 variants. We will now be getting essentially the equivalent of sequence. In the next few years, we will have the equivalent of sequence on every animal. So imagine 20 million variants called on all million cattle in the Angus reference. The challenge now for us as geneticists is how do we use all of that information? Just to touch on genome editing. I'm not an expert in genome editing, but it is really interesting to see what we would consider, 10, 20 years ago as would we see that in our lifetime? The technology is there now to essentially edit the animal's genome, and we can edit in desirable traits. And in this case, it's a red Angus animal where they've introduced the slick coat allele. The slick coat allele comes from other cattle like the Romo Sinuano from Colombia, but we can take that version of the gene, put it into red Angus, and we have a red Angus that's now slick coated like a Romo Sinuano, and they're more tolerant to heat in Brazil. CMEX is working with this company, a press release here in 2018, to basically knock the horns off of dairy cattle, and I've spent a lot of time dehorning beef cattle, and I think anyone who's done it would say they'd be glad to be rid of it. So it's a technology to basically do that. Genetically modified animals are on the plates of consumers in Canada today. This is the Atlantic Aqua Advantage salmon. And those two salmon are basically the same age. The one above, it's been basically genetically modified around growth hormone, and what growth hormone it produces and how much compared to the salmon below it. And I've seen it. It's been a long time, so I remember those pictures from even when I was a student. But that's now available for in Canada. So looking ahead, as Mike said, livestock faces a lot of challenges. I think genomics provides a key solution. In some ways, I see it. It's if you can dream it in terms of what you want to change, you can change it. It's simply a matter of you're going to measure it, you're going to genotype it, select, and then you repeat. Measure, genotype, select, and repeat. The method and the recipe is there. It's really up to the industry to determine where they want to take their what challenges do they want to tackle. And if they can measure it, they can change it. And finally, understand the genome. As I indicated, we're going to go to whole sequence in every animal, but we really don't understand the genome very well at all in terms of what all those individual genes do. Yeah, we can gene edit animals now. Where do you start making edits if you want to improve worm resistance or health in some of these traits? What we've tackled so far with gene editing are pretty simple traits: fold, slick. Those things are controlled by individual well-known genes. These other traits are a lot more dynamic with a lot more mechanisms involved. With that, I'll stop and turn it over for questions.
Thanks, Steve. That we let you go on a little bit because interesting stuff you've got to share, but we do have some questions in the chat box. And one of them, well, the first one is, what are the current usage examples for the poultry industry? How can it help infection and disease resistance?
Yeah, so there was a graphic I didn't include in it. I said so for poultry, the major breeding companies, so I used turkeys there as an example, but whether it's broilers or layers, at the parent company level, they will be using genomic selection to basically improve their poultry. So that's giving them more accurate breeding values younger when they're selecting that parent stock. But to do that, they need to actually measure those traits. So if they want to improve health, for example, in poultry, and I'm not exactly an expert in terms of exactly what are the actual health traits that are being measured, say in broilers and layers and things like that. I know because I've sat through a lot of talks on the turkey side, because Ben gave a lot of presentations at Guelph over the years. They're measuring on the turkeys. They're actually measuring a number of traits that are related to health and welfare. For example, leg strength and other kind of walking ability traits in turkeys are actually measured at the top of that tier level. And then so the genomics is just helping speed up those genetic improvement programs. But the companies need to be measuring those health traits that are of interest.
All right. Is there any interest in the prospect of moving the shedding ability of cattle into sheep in areas where wool has no value?
Yes. So yes. So there are some shedding breeds, your Dorpers, your Katahdins, and things like that. And before I was at American Angus, I was actually in New Zealand with a company there called Egg Research, and some of those breeds, they were very much interested in shedding ability. So you could essentially apply it's the same thing that we applied in Angus cattle, scoring because it is quite heritable, and there was people developing other lines of sheep in New Zealand, Wiltshire, and other ones that were shedding lines. But you can score your sheep for shedding, and with that reference, you could apply genomics just like any other trait. But the shedding trait is actually very heritable. So even without genomics, you can make some progress. The idea with genomics is just going to make faster progress.
So the way to make faster progress is more data.
Yes. Yeah. So the genomics isn't going to help if you don't have the that reference set of animals. And part of the problem with some of the, well, Dorpers are big in Australia. There's lots of Dorpers, but in Ontario, if you don't have, you need thousands of sheep measured for the trait with genomics to actually really make it work.
So the more producers share their data, the more information we'll collect and can utilize. Yeah,
and that's really, I guess, fundamental to like that. What I showed there today, sheep genetics. That is a national system. All the breeders are basically sharing their data through that.
Mike McMorris asked a question. Some people say genetically edited is different than genetically modified, though still many think genetically modified involves transgenics. What's the correct interpretation?
So yeah, so genetically modified, for example, there's I guess the difference is well, there probably is actually. Mike probably knows the actual official definitions, and maybe I'm not exactly up on it per se, but I would say when we go to genetically modified, in the old days, for example, there was you think about whether it's crops, corn or soybeans, they're genetically modified. They're actually taking DNA from another species and putting it into in the crop. And we had a genetically modified pig that was developed at Guelph called the Enviro pig, and that pig actually was able to better use phytase, or better to use the phosphorus in its feed to produce less phosphorus out the back end of the pig, which was better for the environment. But the mutation for that gene in the pig didn't come from pigs. I think it came from bacteria or something. I think that I can't really remember now. That was 20-something years ago. But that was a genetic, we would call those GM. What we're talking about, the interesting thing now is we're talking about gene editing, but we're just, take that Red Angus calf, for example. We took so we have a bovine, a Bos taurus beef animal, and we've basically, we know this mutation exists over here in Romo sinuano, and we could breed Red Angus to Romo sinuano, and we could start to cross back to Red Angus and only keep the ones that kept the SLIC gene, and over a number of generations and a number of decades, we would eventually have a SLIC Red Angus. So all that the gene editing's done, it says, well, why wait, 30 years or something to do all that back crossing? We could just go in and make the edit and have that animal now. And so gene editing is, we've edited a known mutation that exists in other cattle into this other cattle over here. And that's kind of where I see, that's how I distinguish between genome modification and gene editing. I think they're both under the umbrella of genetic engineering. That we have engineered the DNA on that animal.
Okay. Can there be parallels drawn across species so you don't have to have the measurements to verify? For example, feed efficiency in sheep. Can we use work done on beef feed efficiency traits to help select for feed efficiency traits in sheep?
Potentially, yes. In the longer term, there has been some work looking at that particular. Actually, feed efficiency was a trait, and looking in dairy cattle and beef cattle. So there was a lot of work done in beef cattle, a lot of work done in dairy cattle, and some work here at the university, or here in Melbourne in Australia with Mike Goddard's group. They looked at how could we actually use the data from beef to help inform dairy and vice versa. And they did show some increased accuracy there. It is pretty difficult because for the most part, with the technology as we implement it today, for example, if I'm don't pick feed efficiency, pick any trait, say carcass weight, in Angus, say, I've got a genomic prediction for carcass weight in Angus. I wouldn't expect that same prediction to work in Charolais. So we can't just take a prediction in one breed and use it in another breed, more or less across species. But we might identify, say, in beef cattle for feed efficiency, that cattle that are more feed efficient have, a mutation in, say, this gene which causes them to produce. We did some work at Guelph with Kendall Swanson years ago around trypsin. Then maybe they produce more active trypsin, which improves protein digestion. Well, that could apply in sheep as well. So I think there's definitely learnings that will come about across species. As we drill down into sequence, we will identify what are the causative mutations, and we'll see that while there'll be different mutations, of course, in sheep versus cattle, but in those genes, there's mutations that improve nutrient digestion, and it's the same genes in cattle, just different mutations.
Okay, and I'm going to go to a couple other questions. Pretty much, I think you've pretty much answered, but we'll send direct answers out to the people. I am going to make one. There's one last question here. I believe you probably know a guy named Dan Ferguson.
Yes.
And he's got a comment in here, and he says, Zoom is such a great tool to bring Steve back to Ontario. Will all the narrowing of genetic choices lead to a more vulnerable bovine species? Some producers have this theory. What is my Miller answer?
It depends how you use the tool. So there is the potential. It could go both ways, but if this question comes up a lot, Dan, it's good to hear it. But in genomics actually could allow, and we have looked at it in Angus. Have we decreased the genetic base since we brought in genomics? And the answer is no. It's actually has not decreased. And the reason it could actually increase, if we go back to Holsteins, for example, back before genomics, we had sire analysts that they would be kind of almost infatuated with certain cow families. So here's this cow family and her daughters, and those were flush cows, because they didn't have any good information on those cows really. But with genomics now, they can actually have much higher accuracy of selecting on the female side, and they can cast the net a lot wider. So really with genomics actually enables you to cast the net wider instead of being more narrow, because before genomics we had to progeny test bulls to determine who's the best for carcass. Well, we could only progeny test so many bulls, but with genomics now we can actually look at a lot more bulls and basically cast that net wider. So it really comes down to, you could say, well, we're going to identify the best animal and everyone's going to go for it, but with cheap genotyping we can actually get better predictions now in a lot of animals and cast the net a bit wider. So it has the potential to narrow your base, but if employed correctly, and that's actually one of the applications of the technology, I guess, is we want to maximize our rate of progress, obviously, but we don't want to decrease our genetic base. And so there's mating algorithms we can apply to balance those things.
Thanks very much. So I want to thank you for getting up early and talking to us from tomorrow, because I believe it's about 7 o'clock in the morning there.
It's coming up to 7 o'clock. Yep, sun's just getting up. So
that we did, as mentioned earlier, we have recorded the webinar. We hope that if technology is on our side, we'll have it posted by the end of the week. We'll send everybody a link to that. Our next webinar is going to be on June the 28th, and it's going to be back at our normal time slot at 12 to 12:40 with Dr. Steve Roche, and it's on getting research into practice, as you can see and have a look at that. There's a lot of good information in there, and the more everyone provides data into the system, the better it can be. And the paper does a good job of explaining that. So again, Steve, thanks for getting Charlotte, keeping us all in line. And thanks everyone for joining, and hope to see everybody again on the 28th.